So today I want to talk about MHC structure and function. So at the beginning, what I'm going to tell you about is a little bit regarding MHC genetics. It involves some nomenclature stuff that is a pain in the butt. However, you kind of have to know it to understand any immunology that you read that has to do with any of this. And then we're going to talk more about sort of the actual structure of the protein. So it's going to switch into almost like biochemistry after we do a little bit of genetic stuff. If you recall before break, I told you that the MHC genes are the most polymorphic genes in the human genome. So when we think about all of these different MHC genes, for example, we think about this one that's listed as HLA-A. There are 2,980 alleles or variants found among the human population. And so the sort of little first part of this, which is sort of thinking about genetics, is also really a way that we're thinking about what do we call those 2,980 different versions? How do we name them? So if I were to look at your MHC, if I were to do sequencing and try to figure out, which one you have, what does the nomenclature look like for what we call that? And so that's the sort of the first part of what we're going to talk about. And we're going to see the nomenclature in both humans and in mice. As you'll see in a couple slides, MHC was discovered in different species in slightly different ways. And at the beginning, they didn't know they were looking at the same genes and proteins in the different species. And so as a result, there sometimes are different names for the gene locus and the proteins in different species. MHC, which stands for major histocompatibility complex, is the overall name. So MHC applies to every organism with this. But different organisms, MHCs, might have their own unique names. In the case of humans, we refer to the human MHC as HLA. You may very much hear me using MHC and HLA interchangeably, because technically HLA is the human MHC, and I often am thinking about examples with humans. HLA stands for human leukocyte antigen. This is because HLA, the first studies on HLA were actually done looking at weird proteins that were on the surface of leukocytes that varied between pregnant women and their fetuses. Which now you can kind of understand, like, oh yeah, the pregnant woman and the fetus wouldn't have identical, some would come from the dad. So okay, they'd be different. And so it was just like, look at these weird proteins that are on the surface of the pregnant woman versus the fetus. In many organisms, what people were doing was actually transplanting different tissues and looking to see what happened with tissue transplants. And so that was histo or tissue compatibility. And for a long time, nobody knew that the weirdo proteins on the surface of the leukocytes were also the same one as the histocompatibility. So that's why we have two different names. The same thing. Don't get super stressed about it. So if I mention HLA or the HLA locus, I specifically am referring to human MHC. And I told you last time that we've got the class 1, class 2, and class 3 regions of the genome, all on human chromosome 6. The class 3 region encodes some sort of other proteins of immunologic importance that I'm not going to say a whole lot about. But the class 1 region encodes the class 1 MHC proteins. And the class 2 region encodes the class 2 MHC proteins. In humans, the class 1s are known as HLA-A, HLA-B, and HLA-C, while the class 2 loci are known as HLA-DP, HLA-DQ, and HLA-DR. And I like to remember that the ones with one letter after HLA are class 1, and the ones with 2 letters are class 2. We each have different HLA-As from our mom and our dad, different HLA-Bs from our mom and our dad, HLA-Cs from our mom and our dad, like we saw last time. And in humans, the way that we distinguish those different versions of, say, HLA-A is with numbers. So you might have HLA-A1 and HLA-A253. It's basically just a set of numbers, that we use to distinguish different versions of it. And so I will often, talk about them with the different numbers, telling us which version of HLA-A or B or C or DP or DQ or DR, we might have. In mice, MHC was discovered through a completely different set of experiments, and people, like I said, didn't know that they were actually discovering the same genes and proteins in these experiments. And so they couldn't come up with like a sensible name that was the same as in humans, because they didn't know it was the same thing. The way that they described what was happening in mice was by looking at skin grafts or skin transplants between the mice. And typically you do this because of all the tissues, it's pretty easy to get the skin and transplant it, compared to anything else. One big difference that I want you to think about when we think about mouse versus human is that when we are thinking about mice, we're talking about laboratory mice that are bought from the laboratory mouse store and that have been genetically inbred since like 1900, so for more than 100 years. So every mouse of a particular mouse strain is genetically identical, and we have its entire genome known. They're known as inbred lines, inbred species. Basically, somebody in like the late 1800s, early 1900s found mice, like wild mice that had weird traits. And they were like, I wonder what happens if I like breed it together too with the weird traits, if I can like get a true breeding line. And so they did that. And in the end, we got a bunch of mouse strains that we still use now. What I want you to know is that humans are not inbred species. Humans are outbred species, which means that our moms and our dads are not genetically identical, leading to us being kind of, easily genetically understandable. If mom and dad was genetically identical, then we'd actually be genetically identical to that too. That's not what happens in humans. And so there are a few places when we talk about some of the genetic stuff where things will look a little different in humans as an outbred species versus mice as an inbred species. And so here you can see sort of the background of some of these mouse MHC experiments. These experimenters had 2 mouse strains that were true breeding mouse strains. You can see one is listed as orange, one is yellow. You can see that the orange one, it says, has 2 copies of the B genomes, while yellow has 2 copies of the K genomes. And they could breed those 2 mice together to make a mouse that was the F1 progeny that had basically 1 copy of the B genome and 1 copy of the K. Got the B from the B parent and the K from the K parent. And so you can see it's both orange and yellow. And then what they tried to do was they tried to do skin transplants between these 3 mice to see what would work. And this was what they found. Please note, when I tell you this, this is true for inbred mice. Do not go home and tell your parents to give you kidneys based on this, because you are an outbred species. So things are a little different in an outbred species. But what you can notice here is that we've got this B or orange parent. It could give skin to other mice of the B strain. It could not give skin to mouse of the K strain, so that skin would get rejected. And it could give skin to the progeny, to the child. You can see that same thing happening with the yellow K parent. It could give skin to another mouse of the K genetic background. It could not give skin to a mouse of the B genetic background, and it could give skin to the progeny. And if you look at the progeny, it could give skin to other progenies that are both B and K, but it could not give skin to either parent. And so this sort of let them say, okay, there's this gene that seems to be controlling whether or not you can do this skin transplant. Basically, you have to have the gene. So here we know, well, the other B mice have the B gene. The progeny has the B gene. Other K mouse has the K gene. The progeny has the K gene. Here, you gotta have both B and K. Neither of these guys have both B and K, but this progeny does have both B and K. And so they started to do experiments like this, and they were doing finer and finer kind of mapping to find the gene that was involved in this process. And instead of calling it the human leukocyte antigen, because at this point, how do they know there's leukocytes and there's no human? Like, why would you name it that? Instead, they named it the H2 locus. And it was really for histocompatibility. I don't know where they got the 2. And so just like we can see that section of the human chromosome 6 that encodes the HLA locus, we can look at mouse chromosome 17 and find the H2 locus. Just like in the human HLA locus, we have a class 1, a class 2, and a class 3 region. You will notice that in mouse, the class 1 region is actually split. I'm always going to draw it as together on the one end, the same way that human is, just because there's no reason. We don't need it to be split for our purposes, but yes, this image does show it to being split. The class 3 region encodes other genes of immunologic importance. In fact, it's the same ones that were in the class 3 region in humans, like complement and some cytokines. The class 1 region encodes the class 1 genes in mouse. And the class 1 genes in mouse are H2K, H2D, and H2L. In the class 2 region, we have the class 2 genes. And in mouse, the class 2 genes are H2IA and H2IE. So again, my whole thing about 1 letter versus 2 letters holds up here as well. And so one thing that you should sort of notice, you should be able to notice from this is that if I say, H2, if you see H2 in a question, your brain should go, oh my gosh, we're talking about mouse MHC. If you see HLA, you should automatically be like, oh my gosh, we're in human MHC. When I was in graduate school, I did some work with rhesus macaque, and so I can tell you that if you saw the word mammu, which is just a shortened version of Macaca mulatta, the genus and species name of rhesus macaque, mammu is the name for rhesus macaque MHC. There's some weird name for every species. But we're only going to really care about HLA and H2s. The trick with H2 is that they didn't use numbers to distinguish the different alleles. So, like I said, I might have HLA-A, and Brooke might have I might have HLA-A1, and Brooke might have HLA-A17. If we have 2 mice, one of the mice might have one version of H2K, one mouse might have a different version of H2K, but we don't use numbers to distinguish the difference. And this is the thing that's the pain in the butt. This is really based on the fact that mice are inbred species. And so, and the 2 most common inbred mouse lines that I use as an immunologist are Balbc mice, which are these white albino ones, and C57 black 6s, which are shown over on the other side, which is also what you used in lab. And somebody back in the day, I don't know why they did this. It's one of those things that now you're like, what? It's okay. How it is. Said, you know what? All of the MHC it's like, imagine somebody came to me and they said, every HLA in Dr. Barker is the number 1. So she has HLA A1, B1, C1, and also number 2, A2, B2, C2. One from her mom, one from her dad. And that was how they defined it. They did it similarly with mouse strains. So they said every H2 gene in a black 6 mouse is known as the B version. And so they said that the black 6 mouse is of the H2B haplotype. And what it means is when this mouse is of the H2B haplotype, what I know if I see that a mouse is of the H2B haplotype, it means that it got the version of K that it got from his mom is called K of B. The version of K that it got from his dad is K of B. The D that it got from his mom is D of B. The D that it got from his dad is D of B. L of B, L of B, IA of B, IA of B, IE of B, IE of B. And so basically somebody went through and said each mouse strain gets a letter. And all of the MHC types in that mouse strain have that letter. And so instead of saying HLA-A1 or H2K1, we call it K of B. We have this little subscript of B. So this, In the Biol mouse, the white mouse, it is of the H2D haplotype. So H2 subscript little d. And when I see that it's of H2D haplotype, that means I know, that it has 2 copies of K of D, 2 copies of D of D, 2 copies of L of D, 2 copies of IA of D, 2 copies of IE of D. Yep, Brenna.
The reason why that is because then you don't have to distinguish between the parents or genetically.
If it's a strain, I could. So let me give you. So I was going to give you another kind of example, but let me give you that example too. So let's imagine I have my black 6 mouse that is H2B, and I have my Biol C mouse, which is H2D. I could cross them and have a progeny. What I would actually call that progeny is H2BD, like that. And that would tell me that I have this. That I got one chromosome that was all the B ones and one chromosome that was all the D ones from each of my 2 parents. So if I told you, I have an H2B mouse, then you can assume that that's what's going on with both chromosomes, is B. But if I say, like, it's progeny of this and this, then you've got to imagine one chromosome from each parent. This also brings up sort of one other important thing, which is that sometimes we use this word haplotype when we talk about the MHC genes in a particular organism. Officially, the word haplotype means a group of genes that are inherited together. It has to do with how frequently there's recombination between them and how frequently we kind of switch up which ones are on the same chromosome. And so one thing I would be able to tell you is that in this mouse, this set of MHC genes is one haplotype, and this is the other haplotype. Basically, which set of genes are inherited together, because they're on the same chromosome, they're linked, so they have to be inherited together. The MHC locus in humans is the longest haplotype in the human genome. So it's the longest section of the human genome that tends to all go together without a lot of recombination in between it. Sometimes it helps people to understand that if I tell you about the second longest haplotype in the human genome. The second longest haplotype in the human genome is the stretch of genome that includes the skin pigmentation, eye color, and hair color loci. So if you think about it, if you were going to imagine a person who had very, very pale skin, you probably imagine them with blue eyes and blonde hair, right? Those often go together. But you also know that it's not 100%. There are some people who have very, very pale skin, blue eyes, and not blonde hair. Because in the course of the human population, there has been some recombination, but it's just kind of a frequency of how often those genes are inherited together. And so we do think about the MHC haplotype. So what I'm going to do is I want to show you one other mouse example. And then I'm actually going to flip forward in the slides to a couple of problems that I hope will help make this all make more sense. So this weekend over break, I went into the city, and I was riding the subway around, and of course there were lots of mice running around the subway. So let's imagine for a second that I caught one of those mice. I don't know why I caught one of those mice. That seems like a very bad idea, but whatever. Because it's a mouse, I know it has 2 copies of chromosome 17. And I know that it got one from its mom and one from its dad. And so I know that it's got 2 K genes, 2 D genes, 2 L genes, 2 IAs, 2 IEs. I know it's got this. What I don't know about my random subway mouse that I've caught is which versions of those it has. I could, however, do some sequencing of my random subway mouse that I caught. And if I did that, I might find, it happens to have a version of K that's the same as in a black 6 mouse. So it has a K of B. And its other K is the same that's in a Balb C mouse. So it's a K of D. And maybe it has, I found out that it has an L of L, and it has a D of W, and an L of K, and an L of B, IE of D, IE of K. I don't even remember what all the choices are. S. Oh, apparently there's S. S and Q. Okay, great. And so this is what I might find in that random subway mouse that is an outbred mouse that I've just caught. And so the idea here is that we're using those subscript letters just to tell us which version of the gene we have, in the same way that when we look at human, we are using numbers to tell us which versions of each of these genes we have. One of the reasons why this is particularly important is that when you look at the problem set and at the old exams, you will see that sometimes I will list, this is what's up with the MHC of this mouse. And then I'll ask you some questions about what's going on MHC-wise, and you need to be able to read that information that may include information that looks like this and know what's up to use that. But I also want to move, like I said, to show you these 2 problems. So this is just something that came out of one of the immunology textbooks. And here we're looking just at the MHC class 1 genes of some individuals. You can see that we have offspring. You can see that we have biological mother, and you can see that we have potential father. And we can use what we know about these MHC class 1 genes to figure out which of these is the potential father. We can be like Maury Povich. You're not old enough to necessarily know what that means, but that's okay. And in fact, when you think about a lot of these kinds of paternity things, sometimes this is what they'll do. If you think about some crime scene investigations where they're like, there's a 1 in this many billion chance that it's that person, it's because they looked at the MHCs and saw if the MHCs matched. So if we look at these data, which one of the potential fathers is in fact the father of this offspring, and how do you know? What do you think, Brian? So Brian says number 2. Why is that? Okay. Okay. So if we look, all right, so this child has A3 and A43. It had to get A3 from its mom because mom didn't have 43 to give it. So that means 43 had to come from the dad. The only dad who has 43 is number 2. And we can actually go through and check the same thing with B and C. So this child had to get B59 from its mom, so it must have gotten B54 from the dad. So that only lets father 1 or father 2 be the choice. So father 2 still works. This child had to get C8 from mom, so it had to get C5 from dad. So again, possibly number 2. And so that helps us figure that out. And so this is sort of the stuff you can do by thinking about some of these MHC genes and the way that you might think things through. But this is another, this is actually a question I asked at some point in the past in my life, where we're looking at this. And what I want you to notice is that in this, so we're looking at a family, father, mother, child, and you can see that specifically in the question, I'm asking you for the MHC haplotypes of the mother and the father in this family. What that means that I want is I want you to write 2 chromosomes for each person. I want 2 chromosomes for mom and I want 2 chromosomes for dad. Because I want, as I said, both haplotypes for each of them. And so what we can do here, and in fact, this is really what would have happened in that, what I did this with my subway mouse too, is I can do sequencing and I can find out these are the HLA A's, B's, and in this case, DRs of each of these individuals. But what I don't know is which A is on the same chromosome as which B and the same chromosome as which DR. Like, which ones travel together from one person to another. That's what haplotype means. It's a bunch of genes that are traveling together. And so we can think about this as what is one of mom's chromosomes? What's the other of mom's chromosomes? What's one of dad's chromosomes? What's the other of dad's chromosomes? Does anyone want to start out by giving us one of mom's chromosomes? Yeah, David.
A2, B27, and DR3.
And how did you get that, David?
Because those 3 were the 3 that were passed to the child, so they probably, or they work on one chromosome.
Yeah, so we know that mom gave the child A2, B27, and DR3. And so those must have been attached to each other on the same chromosome. So, who wants to give us mom's other haplotype? Mom's other chromosome. Yeah, Brenna.
A3, B12, and DR3.
So there's mom's second haplotype, mom's other chromosome. Those 3 genes must be on the same chromosome because they're not on the first one and she's only got 2 chromosomes. So those are the leftovers. All right. So now we need to figure out what is dad's first haplotype. Yeah, Brian.
A20, B57, DR5.
And we know that because those are the ones that dad gave to the child. So that those 3 have to be together on one of his chromosomes. One of his versions of chromosome 6. Which 3 are on his other version of chromosome 6? Yeah, David.
A1, B7, DR2.
A1, B7, DR2. And because those are the ones that are left. And so they must be sitting on the other chromosome. And so this is the way you would address sort of one of these questions to understand haplotypes in a family. And part of this is expecting or hoping that you understand this term haplotype of a bunch of genes that are on the same chromosome that are linked and are inherited together. All right. So that was my discussion of MHC genetics. And now we can start thinking about MHC function. So for the rest of the time today, we're really going to be talking about this MHC molecule here, that is presenting a peptide, kind of what the structure of it looks like, as well as a few other important features of this protein. Then starting on Friday, we'll talk about what's going on inside of the cell to get that peptide onto the MHC molecule. In order to talk through all of the details of MHC function, I have to give you a quick reminder of a couple of things about proteins and protein structures. Hopefully this is not totally novel information for you. So one of the things that we're going to need to know here is that when we think about proteins, we should remember that they are composed of amino acids, all bound together. And you can see two individual amino acids at the top. You can notice that one of them has, that on one end they have this N, either H2 or H3. That's the amino group. Then we've got sort of this carbon in the middle, and then we have this COOH group, this carboxyl group. Okay? And we're going to then bind a carboxyl group to the amino group of the next protein. And you can see that here, you can see a longer amino acid here. And so what you can see is we've got this nitrogen from the first amino acid, carbon-carbon, nitrogen, carbon-carbon, nitrogen, carbon-carbon. It's like long string of this. Often the nitrogen at the very end, at the amino terminus of the protein, will actually have a positive charge. So you can see it's actually an NH3 in this case, while the oxygen at the very end of the chain will have a negative charge, the carboxy terminus. You can also notice that off of the central carbon, we have something that's listed as an R group. You can see those R groups that are shown here. And I'll say a little bit more about the R groups on the next slide. But the big thing that I want to sort of define for you based on this information is that one thing I'm going to talk about a little bit when I talk about MHC proteins and sort of what's going on here is sometimes I'm going to talk about the main chain atoms of a peptide. When I talk about the main chain atoms of a peptide, I could also maybe say the backbone. When I talk about the main chain atoms or the backbone of the peptide, I mean this string of NCC, 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 NCC. That's the main chain or the backbone. No matter what protein we're talking about, the backbone or the main chain always goes NCC, 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 and on and on and on. So no matter what protein we're talking about, the backbone's always identical. But when we also think about our amino acids, we know that there are some things that are not identical about them. We know that there are 20 different amino acids. And the place that those 20 different amino acids differ is in what's on this R group here. And so we've got lots of different R groups. And so if I mention the R group, that's something that is going to differ from amino acid to amino acid. And the R groups that are present will change from protein to protein or peptide to peptide, while the main chain will not. You can see the 20 R groups here. No, I'm not going to ask you to memorize the 20 R groups. That's biochem. One thing I also will point out is that they have 3-letter abbreviations and 1-letter abbreviations. I'm often going to use 1-letter abbreviations, You don't really have to know them all, though I'm guessing by the time we're done with the semester, you'll know why is tyrosine pretty well, because I use tyrosine as an example all the time. But what I want you to notice in looking at the R groups is that they vary a lot in shape, they vary a lot in size, and they vary in charge, as well as sort of if they're hydrophobic or hydrophilic. And so you can see we've got here's one that's like long and skinny and that also has a positive charge at the end. Here's one that's really tiny and doesn't have a charge. And so what you might imagine is that different R groups in certain situations are going to bind to sort of pockets that match their shape, and charge. So I told you guys that I've told people in the past that as an immunologist, I mostly think about whether or not shapes match. And so here we're going to think about the shape of the R group and whether or not it matches certain pockets. All right. So, I've already told you about MHC genetics and the different MHC regions, the class 1 and the class 2 region. And oh my gosh, they encode different kinds of proteins. The MHC class 1 region encodes MHC class 1 proteins, while the MHC class 2 region encodes MHC class 2 proteins. And these two types of proteins have somewhat different structures and a bunch of different important properties. And so I'm going to tell you specifically a bunch of stuff about MHC class 1, and then I'll go back and tell you all the stuff about MHC class 2. And so that's another thing you should always be paying attention to when we're thinking about anything regarding MHCs is, are we talking about class 1 or class 2? Because the information is going to be different for them. And so, when I say HLA-A, I know to myself human MHC, and I know it's class 1. And so those are kind of quick things that you want to be aware of. If you remember, there's a question that Jonah asked before break, and I sort of had to hedge my answer to it, and the real reason is because the answer is different between class 1 and class 2. And in fact, I'm going to specifically address exactly that information as one of the things I talk about with class 1 and class 2. So here you can see MHC class 1. MHC class 1 involves two protein chains. One of those is known as the heavy chain or the alpha chain. And that, you can actually see it from your textbook here. These 3 domains in yellow. That's the alpha chain or the heavy chain. And that is actually the part that varies between different MHC class 1 molecules. All MHC class 1 molecules also pair with a partner protein. The partner protein is the same. So you only have one gene for this partner protein. That you make a bunch of copies of it, and they pair with all your different MHC class 1 heavy chains. You can see the partner protein is shown here. What I want to point out is when I've asked people to draw it, one thing that is really important is that it's not covalently bound to any of the rest of the MHC molecule, and also it does not have a transmembrane domain.
I just want to make sure you said the alpha chain is what differs in the class 1?
Mm-hmm. Yep, and so you can see this here again. We've got our alpha chain that's shown here, and then this partner protein, beta-2 microglobulin, or beta-2M. Beta-2M does have an immunoglobulin fold structure, as does the alpha-3 domain, which is the transmembrane part of class 1. The alpha-1 and alpha-2 domains, you can see they're not drawn with that nice semicircle structure here because they're not perfect immunoglobulin domains. And alpha-1 and alpha-2, so alpha-1, alpha-2, come together to make the peptide binding cleft or the peptide binding pocket. Alpha 3 is sort of this structural part that's holding it up and has a transmembrane domain. And then beta 2M comes in as this final piece for MHC class 1. This molecule is not folded correctly unless it has MHC class 1 heavy chain, beta 2M, and the peptide. So you need all of them to actually put this whole thing together. That's going to be really important when we talk about how exactly you get a peptide, because it doesn't fold right until you got a peptide. One thing that is actually also really important is that there is sort of an on-off rate biochemically of beta 2M. And so when it comes off, the peptide actually comes out of the cleft. And so what really we think is happening with class 1 is that beta 2M is coming on and off, and it's like it's throwing and catching the peptide as the whole thing like breathes on, off, on, off, on, off. And I point that out because you can actually use it for some kind of cool experiments based on that. So you can see again that the alpha 1 and alpha 2 domains are coming together to make that peptide binding cleft. So it's the same protein that's making the peptide binding cleft. You'll notice that there are alpha helices on either side of where the peptide goes, and then there's like a beta sheet underneath, like holding it like a little platform. Right now we're looking at the MHC class 1 molecule from the side, but we can also turn and look at it down like we were the T cell. And if we looked at it like we were the T cell, this is what we would see. And so what you can see is that we've got our two alpha helices, one coming from the alpha 1 domain, one coming from the alpha 2 domain, and they come together and they sort of give us this little area where the peptide can be bound. And so here you can see that peptide being bound. You can see that here as well. And I really like this image. This is a space filling model of the atoms, kind of like what it would look like as a surface. The white is the alpha protein, the heavy chain, and the orange is the peptide. The blue is the beta chain. And so you can see it's really one big surface of alpha 1 and alpha 2 coming together with the peptide. There are a couple of key things that I want to tell you about this peptide binding cleft. One of them is actually shown here, even though you didn't know it was shown here. Because you haven't seen class 2 yet, so you don't know the comparison. If you look at this peptide binding cleft, these parts right here at the ends, you can see are white. They're made up of the alpha chain. This binding cleft has a very specific length of peptide that it will bind. It can't bind peptides of infinite length. It's got a pocket that's really specific in how many amino acids it can bind. And so this peptide binding cleft will bind peptides that are 8 to 10 amino acids in length. It might be 8, it might be 9, it might be 10. So like, I don't know, I'm making this up. K of B could bind 8s. K of D could bind 10s. HLA-A1 could bind 9s. I don't know the numbers for all of them. But they all, MHC class 1 either binds 8 amino acid long peptides, 9s, or 10s. So it's really, really strict in that way. Because the ends of this cleft are closed. Usually this part of the cleft, these amino acids right here, have a negative charge because there's going to be the positive charge from the N amino terminus here. And so it's going to help attract the N terminus of the peptide. These over here are going to have a positive charge to attract this negative charge. So there's my cleft. You can see that it has a defined length. I'm only going to be able to fit so big of a peptide in there. And it has, part of the pocket has these negative charges to interact with the positive here. Part of the pocket has positive charges to interact with the negatives here. So that's one kind of key feature about the MHC class 1 peptide binding cleft. There's sort of one other really key feature about the MHC class 1 peptide binding cleft. This, Jonah, is your question. Which is that the MHC class 1 peptide binding cleft interacts with R groups. This is the version from your textbook, but I'm also going to sort of draw this and show you a couple of other versions. And so what this version from your textbook shows is that if we look at our peptide in the peptide binding cleft, and we look at the actual biochemical interactions between the cleft and the peptide, those interactions are all with different parts of the R groups. And you can see this here as well. So you can see that some of the R groups will be pointing down and interacting with the cleft. And they will have a specific shape and charge of whatever that R group is. And you can see some of those R groups will also be pointing up towards the MHC molecule. And so really, if I were to draw my class 1 correctly, I would draw, I'm like, maybe here it has a long, skinny pocket with like a negative charge at the bottom. And maybe over here, it's got like a bulky pocket. And so it's going to fit peptides that are 8 amino acids long that have something that can interact with this pocket and something that can interact with this pocket, based on the R groups of that peptide. And so you can see that here. Those individual amino acids that are interacting with the MHC class 1 peptide binding group are known as anchor residues. And so what you can see, there's two examples here. If we have MHC class 1, it's only going to bind peptides that are 9 amino acids long. And it can only fit peptides that have a G at position 2 and a P at position 3, and either an I or L at position 9, based on the sort of the shape of its pocket that will fit in. While a different kind of MHC class 1 might have a pocket that fits tyrosine at position 2, so kind of a large, bulky position 2, and valine or isoleucine at position 9. At anchor. I said anchor residue. Hold on. Anchor residue. In fact, the way that this was shown is that somebody actually went through a bunch of cells, sheared off the MHCs, and sorted them into like type 1, type 2, type 3, and then pulled the peptides out and sequenced the peptides, and showed that like, oh, type 1 binds anything with a G at position 2, a P at position 3, and an L or I at position 9. The people who actually did that experiment, this was an undergraduate thesis that did that. And so for any particular MHC class 1 molecule, we can name anchor residues. So in order to bind to HLA-A201, you must have an L or an M at position 2, a V at position 6, and a V or an L at position 9. Any peptide that matches that criteria, like this HIV peptide, can be presented by HLA-A201. While HLA-B27, it must have an R at position 2 and an R or a K at position 9. So any peptide that meets those criteria, like this influenza protein, will get presented by that MHC. And here you can see two examples of mouse MHCs, a K of B and a K of D. K of B really likes to bind 8 mers. This 9 mers snuck its way in, but it's usually 8 mers. K of D is usually binding 9 mers. And K of B really can bind anything that has an F or Y at position 5 and an L or I at position 8. And so it happily can protect the mouse against hepatitis B, influenza, LCMV, vesicular stomatitis, Sendai virus, all these different viruses, as well as chicken proteins that we like to use in immunology. While you can see that K of D allows for presentation of different peptides that have a different motif, different anchor residues. Yeah.
Just to clarify, the peptide sequence, that's from the T cell specifically?
Great question. The peptide is from the infected cell. So we are looking here at the infected cell, which is presenting the MHC, to show to the T cell. So it is showing this to the T cell. So we're learning about kind of the structure of like the display case. Yeah, Jonah.
Are the MHCs only present in infected cells?
That's a great question, Jonah. Why don't you wait a couple slides? So as is, actually, honestly, really relating to both questions that you guys have asked, there are some other key things we need to know about MHC class 1. One of them is that MHC class 1 is getting its peptides from the cytoplasm of the infected cell. So, Juliana asked about who is doing this. It's the infected cell, and specifically, these peptides are coming from the cytoplasm. So we're doing surveillance of the cytoplasm with MHC class 1. Another thing that I just have to briefly tell you is that the T cell receptor kind of sucks in that it has very weak binding for MHC plus peptide. And so if you're going to actually get a T cell to bind to that infected cell, you need some helper proteins. MHC class 1 always works with the same helper protein. So it needs this particular helper protein to help it bind to the T cell. And that helper protein is CD8. So if you notice, here is our infected cell, our target cell. It has MHC and peptide on its surface. It is binding to a T cell who has a T cell receptor on its surface. And that T cell receptor is binding to the MHC plus peptide. But in order to stabilize that interaction, if it's MHC class 1, we have to have this additional protein CD8 that is going to stabilize the interaction. CD8 you can see is binding to this alpha 3 domain in sort of the back of the class 1, which is conserved so it can bind to every MHC. So MHC class 1 specifically goes with TCR and CD8. When I told you about T cells, I told you that we got different kinds of T cells. One was called a CD8 T cell, and one was called a CD4 T cell. MHC class 1, I just told you, has to interact with this protein CD8. And so MHC class 1 plus its peptide is what's used to turn on a CD8 T cell. So MHC class 1 turns on CD8 T cells. That means CD8 T cells only see peptides that were from the cytoplasm, because that's where MHC class 1 gets its peptides. And we're going to turn on CD8 T cells. If you were to imagine a peptide that's in the cytoplasm from a microbe, what's the most likely microbe that you might imagine in this case? Who has its proteins in the cytoplasm? Yeah.
A virus.
A virus. Viruses, if you recall, do a lot of stuff inside the cell in the cytoplasm. So the most likely peptide is going to be a viral peptide. And in fact, if I give you a random virus on an exam, I probably mean class 1 and MHC class 1. What do you do? What's the best thing to do about a virus infected cell? We saw this a little bit when we talked about the antiviral state. What can we cure that cell? No, what can we do to it? Kill it. Guess what CD8 T cells do? They kill. So again, this is all kind of matching together with the different types of biology that we see here. And one last thing I want to tell you, which cells of your body do you think should be able to display things on MHC class 1, especially if MHC class 1 is really important for viruses? Which cells do you think it would be? All of them. Actually, almost all of them. There is one kind of cell that does not need to present on MHC class 1. That is not going to have viruses infect it. That one kind of cell is a red blood cell. Because if you remember from lab, red blood cells don't have a nucleus. Since they don't have a nucleus, they're kind of useless for viruses. But all nucleated cells, every cell of the body, expresses MHC class 1. So right now, every cell of your body has 2 copies of HLA-A, 2 copies of HLA-B, 2 copies of HLA-C on the surface. You can ignore the class 2s on this cell. Every cell of your entire body right now has all of these class 1s on its surface. So that answers your question for class 1, Jonah. Every cell of the body all the time, class 1. That will be different for class 2. And we will start with class 2 on Friday.